Góðan daginn, þetta ekki formleg málstofa. This is not a formal málstofa. I just invited Áki Jarl here because we were talking and he was doing a lot of interesting research and has been doing a lot in the past, which he will introduce himself. And it was supposed at first to be quite a small kind of talk, but there's so much interest among our group, and I know externally as well that we decided to actually just stream it because he was happy to have it streamed, and there's also going to be a recording on YouTube. So I hope that works. Otherwise, Áki is going to introduce himself and give us an idea of what he has been doing in the past and what he's interested in doing in the future. Thank you.
Thank you very much, Run. So yeah, on that note, I'll be as informal as I can, and looking forward to increasing my social media presence with the live stream. But yeah, so sort of on that note, I titled this talk Sea Change, which is not maybe the most easily understood word, but it is a word in the English lexicon, which has a source like so many words to Shakespeare. So like any good research talk, I want to start with some classical literature, but this is where that term originally comes from. But generally what it refers to is a massive change, something sort of fundamentally shifts and changes things, and just sort of to keep in mind that nothing in the ocean stays fixed. It changes everything in it. And on that note, I wanted to talk a bit about the past, present, and future of marine life, to just start at a very simple point, and sort of go through how each one of these methodologies and different systems that I've been using with these methodologies have sort of shaped my perspective on the future of marine life, especially around the seas here in the Arctic waters and the near polar waters. And I wanted to start by focusing on this idea that as much as we try to look to the future and predict what's going to happen, there will always be interesting exceptions. This particular photo of a sea urchin I took on the big island of Hawaii in 2017 was snorkeling by a pretty lovely little bay, looking at some coral reefs. And this is not to make you jealous. This is just to point out that this is the bay where I was snorkeling and taking these photos, Kapoho Bay, which next year looked like this. So you see that giant flat black area on the right-hand side? That is the bay, or was the bay. So the bay, the reefs, everything in it. I'm sure this was a particularly fast-moving urchin, so it probably made a good escape. But that bay is no more, given that recent eruption. So regardless of how we attempted to predict what exactly was going to happen to that ecosystem, would have been for naught, except if we had a pretty good error factor in our equation. But on that general note, I'm basing this talk sort of around a concept that was originally published back in 2005 by Brian Bowen, who is still at the Hawaii Institute of Marine Biology, and Joe Roman, who is, I believe, still at Harvard. But they tried to sort of co-opt a series of Nordic historical iconographies in discussing conservation biology, and used some horrible Icelandic to try to explain how in the modern systems of conservation biology there needs to be a cognizance, a consciousness of the past, the present, and the future in all its forms. And so I'm going to structure this talk specifically on the methodologies and the information we gather from looking at the past, here sort of represented by phylogenetics and phylogenetic approaches, as well as the present and the future, which I'll talk about soon, and you'll see I'll give him little corrections on his spelling along the way. And I need to begin on the most perfect model organism for marine evolution which exists, which is the sea urchin. They have a gargantuan effective population size, so they're a pretty good model for that sense. They have effectively random reproduction, so they sort of appropriately fulfill sort of key facets of the Hardy-Weinberg equilibrium expectations, which most other systems tend to violate. Not to be biased, but I did my dissertation on urchins, but they are a really good model. They've also been a developmental biomodel for over 100 years, so we know quite a bit about their molecular biology as well. And we have also got a pretty decent amount of sequence data. So sequence data is at this point kind of the gold standard that a lot of us have to contend with when we're thinking about ecology and conservation projects. So one of the easier things to get quickly is mitochondrial sequences and mitochondrial genomes even, if we're trying to use genomic approaches. So using a mitochondrial genome, I was able to sort of quickly and easily build a phylogenetic tree of various other mitochondrial genomes. In most sequence events, if you're trying to do full genomics, the one thing you're almost guaranteed to get at pretty decent coverage is the mitochondrial genome. So it's one of the easier things to get representative across multiple different taxa. Even if there's not a good reference genome, if there's not a lot of whole genomic sequences, you can pull out mitochondria from a lot of different taxa to compare, which is what I did here across the Strongylosyntrotidae. We have a representative here in Iceland, the Strongylosyntrotus drobachiensis, also called the skollakoppur, or the devil's chamber pot. I'm not exactly sure what it did to earn that name, but it's perfectly edible. It's there's another edible urchin here, the Echinus esculens, which is called Mariagull or Maria's gold. For some reason, that one was the preferred urchin. I don't know what quite explains that. But this is the urchin I focused on, which is a pan-tropical sea urchin called Trinusis gratilla. And this is sort of an outgroup in the same superfamily Odontophora that I focused on here. And so all of this is just to show that from a phylogenetic approach using mitochondrial genomes, using some calibrations of the tree, the blue here is a fossil calibration, and the red stars are a series of molecular calibrations from previous phylogenetic studies, was able to essentially route this tree onto actual paleontological data and try to get a sense of when these massive distributions in sea urchin diversification happened. And surprise, surprise, a lot of the diversification seemed to happen around the end of the Eocene, beginning of the Oligocene, so roughly 30 to 40 million years ago. And what happened right around then was a pretty significant change in the marine environment. The opening of the South Seas Passageway, the increase in ice sheet coverage in the Arctic, and a pretty impressive drop in global average sea surface temperatures, almost 4.5 degrees Celsius, it was estimated. So huge changes in the ocean kind of corresponded with rapid diversifications within the Echinometra, or the Echinodermata. And this was seen across multiple different taxa, but this is sort of the past perspective that we can glean based on evolutionary understanding, molecular understanding. We can glean interesting evolutionary histories if we're really interested in what happened 30 to 40 million years ago. If we're interested in sort of looking at signatures of selection as well, there are ways to do this looking at the past and these tree approaches, obviously. So using a branch site likelihood ratio tests of positive selection, I was trying to look at multiple different genes within the mitochondrial genome and see if any of those genes exhibited clear signatures of having undergone strong directional selection. So as we saw, there was a large amount of change in the ocean environment. There was clearly environmental pressure. This is just the mitochondrial genome, so if we were looking for clear signatures of overall adaptation, we wouldn't find it necessarily there, but I was hoping that maybe we could see some signatures that certain genes may have come out of this not unscathed by neutral variation. But the only gene within the coding regions of the mitochondrial genome across all those different urchins that I looked at, there was one gene that sort of vaguely showed maybe a signature of slight directional selection. So not a great way to find if the environment was driving anything in particular. Both the data was not ideal, but the system is quite good for overall looking at how the environment shapes these diversifications. So moving along now to something in the present. So this says here ecology, and I'm mostly focusing on molecular ecology, trying to actually assess what is happening currently with populations and meta-communities. So I started my career, if you can call it that, looking at Bryozoans, little tiny encrusting marine invertebrates, and particularly a species called water cypera, which you can see by the green dots is distributed pretty widely across the globe. And this animal right now is so distributed and was identified so late in the game, we actually don't know its point of origin. It was assumed to be southeastern Australia because that's one of the first places where it was described. But honestly, it was been found in collections dating back a few years in various different places and seems to be popping up all over the place. So we're assuming it's invasive from there, but honestly, we just don't know. And one of the things we just don't know is what sort of genetic diversity these guys have. So one thing I did early on was to sample some individuals from Northern California bays and try to get some sort of genetic baseline for, are there different types of these that are distributing around the world? And we actually designed a little thing that didn't require sequencing. So in order to save monitors and people some money, so they don't have to spend their hard-gained research funds sending off for sequencing, you could run a single PCR test and run these results in a gel. And whether you got 1 or 2 bands would tell you which particular subset of these Bryozoans you had. But these types of population divergences require really good data to actually get a good sense of what's happening in the here and now. So this was my study system for my dissertation, the Trinusta system. And it was originally defined by Ernst Mayers, that sort of currently there were clear biogeographic barriers that define these populations. And it should be easy to understand the distribution, the current ecology, everything that was going on with these animals, and in fact, how they speciated. That was a big topic of Mayer's, based on understanding the ecology and the biogeography. So he defined the Isthmus of Panama and the Eastern Pacific barriers as sort of the key regions that were keeping these populations diverged. And the first foray into actually looking at the systems from a molecular perspective yielded this map. So a black box here means almost exactly that there is no real divergence or diversity seen from the mitochondrial data sets that were gathered. The slightly textured boxes on the right in the Caribbean and in the South Atlantic, those were slight diversifications in the mitochondrial genome. But the conclusions from this early research was mostly that the Mayer was wrong for the most part when it comes to the Eastern Pacific barrier. Biogeography doesn't really inform much difference. The molecular data suggests that this is all pretty much one panmictic population. So if we change our approach a little bit to the data and maybe start looking at, as a history lesson, microsatellites, if anybody remembers those. The microsatellite data actually suggested maybe there was a little bit more divergence that was going on that better explained the different populations we were seeing across this massive area. And we were able to see a pretty clear signature that in the dark blue here on the far right, those Eastern Pacific animals were pretty distinct from the rest of the Central Pacific and the Indian Ocean. And then once we actually expanded that further and started looking at representative genome data, so we used targeted sequence capture to pull small fragments of representative genomes from hundreds of individuals, we actually saw a slightly clearer diversification of populations. So defining here sort of broad biogeographic areas of East, Central Pacific, and then Indian Ocean and Red Sea, those populations come out pretty nicely. The Caribbean here is actually a different species, so this was sort of just a test to make sure that we were pulling apart representative loci within the genome. And then when we look a little bit closer at a higher level of ordination here, the second and third principal components, we actually see that there are some pretty strong signatures even within the Indian Ocean. You can see sort of the Red Sea in purple stretching out there on the bottom left, the Seychelles in South Africa and Sodwana in Western Australia sort of clumped together in the Indian Ocean. And then the Central Pacific and the East Pacific clustered together by themselves. So that's all well and good, great. We can, if we have the right type of data, we go bigger and bigger, we go from mitochondria and microsatellites into genomic data, we can start to pull apart maybe different populations as they exist and they make sense with more biogeographic barriers. But one specific point of interest with this data set was looking at what parts of the genome are actually then diverging. We've got the population story a little bit more down now, but then if we look a little bit closer at the genome, one of the things we see that when we do outlier detection, the outliers are the immune system genes, environmental response genes, and fertilization genes. So these outliers that essentially tell this strong biogeographic story are not just the sequences that we targeted that we know for a fact to be neutral parts of the genome. We actually see some pretty important coding regions of the genome that are telling us this biogeographic story. And these are functional, highly important genes. So this takes me to the last part here. So I'll give Brian this. Skuld is the one thing he actually wrote correctly. So he gets a 1 out of 3. So it's still failing, but he got at least one of them right. So I'll give him credit for that. So looking now to the future, we can understand pretty well from phylogenetics what sort of deep histories of different populations and species, how they can be reconstructed and what information we can glean from them. We can define different populations as they exist currently, both in terms of neutral divergence and how we define populations and population substructure. But if we start to look ahead, try to think about how do we use this information to actually inform our behavior and research right now, to actually start changing our glances from what's in the past to our own navel to actually forward into the future. And to do that, I really had to work with data sets that I was very unfamiliar with, environmental data and chemical analysis. So I won't pretend that I have great understanding of aragonite saturation and how it's properly calculated with acidified data sets, but we were able to work with a colleague at the National Oceanographic and Atmospheric Administration in the US, Li Qingjiang, who helped us with the chemical analysis and the actual calculation of the different ocean chemistry that we needed. And we started looking at the global ocean environment broadly, because the underpinning of all population divergences is going to have to respond to some extent to the environment and to the local ecosystems at which they're a part. So just to look here on the far left-hand side, we're contrasting across different sea surface temperatures on the x-axis, different levels of pH. And the two different colors are comparing those levels in the 1800s, as we have projected them to be, versus now. Well, by now, I mean 22 years ago in 2000. But so, now in model projection times right now is going to have a pretty good standard of error. But yeah, the now is 2000, let's say that. And so the way we're looking at it is that there's some level of overlap between how we imagine the ocean environment was in the 1800s versus how it was in 2000. And so there's some combination of environmental factors that have disappeared. So they were present in 1800s, they're not present in 2000. And there were some environmental combinations that are going to be present in 2000 that didn't exist in 1800. So essentially novel combinations of environmental factors. If we look at different projections of sea surface temperature, pH, and aragonite saturation, we can start to see under different climate change models, so RCP 4.5 and RCP 8.5. We didn't bother with RCP 2.6 since 2015. We can be pretty sure that that is a goal that will just not be met, and we just have to accept that we're not going to get there. So best case scenario now is still about 4.5. Worst case scenario here is RCP 8.5, which is the business as usual model, essentially unmitigated continual greenhouse gas emissions and all the resulting ramifications of that. And we are looking at the number of disappearing combinations of environments in the sea versus emerging novel combinations of environments in the sea, essentially potentially new adaptive niches that might be coming about in the next 100 years. Well, at this point, it's 2020, so 80 years. So mapping this onto a projection of the world, we use a sigma dissimilarity index, which is essentially just telling us how many standard deviations away from the modern distribution, the variance that exists currently, we're expecting these combinations to go. So when we compare the 1800s to the 2000s, both in terms of disappearing and novel climates emerging, we don't see any extreme cases. Extreme cases here we're defining as a sigma dissimilarity index of 4 or greater. Moderate changes we're defining anywhere from 2 to 4 sigma dissimilarities, or 2 to 4 standard deviations away from the variance that we would expect. So now what we're going to do is to compare the modern day, the 2000s, to the two different climate change models, RCP 4.5 and RCP 8.5. So starting with RCP 4.5, this is, again, this point, kind of our best case, honestly, scenario. We're seeing that a number of extreme differences in combinations of environmental factors are going to be exhibited throughout the tropics. It's a little bit hard to see on these projections, but the Arctic as well is going to be seeing a fair bit of the extreme dissimilarities, projecting out to 2100 under this climate change model. And if we go under the RCP 8.5, we see that about 95% of the world's oceans will undergo something that we would consider an extreme difference from what the current combinations of environmental factors is. Keep in mind, this is based on 3 measured environmental factors that we had ready access to from across the globe. That was sea surface temperature, pH, and aragonite saturation state. So for anything that is actually building a shell using aragonite in the tropics, say corals, that's going to be a pretty bad scenario. Essentially, the combination of factors is going to be both gone from what they're currently used to and completely different from what's ever existed on Earth in the last 400 years. So that story right there is not bad if you're in the temperate north. So the temperate north right now has so much variability that it is one of the only places on Earth where there seems to be comparative stability. But it'll be perfectly sandwiched in between a gigantic tropic envelope that has completely changed and an expanding polar envelope that will be very different, exhibiting some of the highest levels of novel environments that we've never, a combination of environmental factors that we've ever measured. So a lot of focus has been on what can we do to inform conservation and evolutionary biology and stock maintenance, given that we know that the environment is going to change in the future. And I wanted to talk real quick about a particular approach that I've spent a lot of time on trying to validate, and our paper just came out this year. And this is the gradient forest approach. It's a machine learning algorithm, but that's just a kind of a meaningless buzzword. So it's an algorithm that is able to take in information using both environmental data and genomic data, and tries to draw something like a cumulative importance curve, where it says that each individual variant within a genome is this tied to an environment or not tied to an environment. This is not a genome-wide association analysis, so it's not looking at correlation explicitly. It is essentially using a random tree approach to find stepping stones where certain changes in SNP frequency correspond to changes in measured environmental detail. So we're going to use this cumulative importance data to generate something called a genetic offset. So essentially, if a population A here were to exist at this point A prime here, would we expect there to be a large genetic offset, or would we expect there to be a small genetic offset? Say, for example, if population B happened to suddenly be living in an environment where population B prime is. So the idea behind this is that we can start predicting in a local environment where we know exactly what the environment is like. If we've measured enough in that current place, we can sort of use these climate models and say, looking forward in time, will this environment right here be roughly the same? Or if we're looking forward in time, where will this environment be in the future? These combinations of environmental factors, will they exist somewhere on Earth? Essentially, are they disappearing or are they just shifting somewhere? Is the tropic shifting further north? Is the polar vortex essentially, or the polar north going to cease to exist and we'll have a similar environment in the Antarctic? So it won't be gone, it'll just be almost inaccessible to everything that lives in the Arctic. And then we will also predict reverse offsets. So if we pick out an individual plot of land or point in the sea, ideally, and want to know where will locally adapted populations be able to survive in however long, based on our model, where on Earth would that be? So that's sort of the trifecta of ways that we can use this data, both to see how well things are going to do in their current environment, see how well other things are going to do in that environment in the future, and then see where else can these guys go if they need to move somewhere else. So very quickly, I'm going to take you through 3 research questions we use to try to address with this particular assessment. Is neutral variance that we can find having a big effect on this? Does the gradient forest work as a predictive tool, actually, if we are trying to look for where things will be more fit? Does the overall magnitude of gradient forest actually inform that? And then finally, say we don't want to use gradient forest, say we want to use FST or environmental distance, does it matter if we're using GF gradient forest or if we're just using some of the more tried and true approaches? And I used a evolutionary simulation framework called SLIM. This is a forward I'm using the word forward a lot, but this is not a coalescent-based simulation program, so it's not looking backwards in time. It actually allows you to set up a simulation and run it forward in time. So it's quite useful if you want to get especially complex scenarios that allow for all sorts of variations in recombination rates and such. And we set up a little metapopulation within this simulation framework, a 10 by 10 metapopulation matrix. And we essentially looked first is if we had no parts of the genome that were actually adapted by the environment, but we had different environments, would just changing the population size give us different inferences from this gradient forest approach? And we sort of assumed that as population sizes increase, there's sort of more variability within the genome. We probably would expect to see increased GF offset. And we generally did see that when you go from a small population compared to a larger population, you get a little bit of an inflated GF offset overall. So this was sort of the first thing to keep in mind when employing this particular model, is that if you're comparing parts of a metapopulation where there might be few individuals to parts where there might be a large number of individuals, we are going to expect to see a little bit of inflation there. And also that, whether it's increasing or decreasing, we see similar levels of correlation to this gradient forest output. So we do see that smaller population sizes will impact this analysis a little bit, but moving forward, when we're actually looking at the efficacy of this approach to predicting actual evolutionary change, we used a quantitative genetic model where we're actually looking at a complex environment. So we're looking at two different environments at the same time, overlapping each other. So I'm showing them split up like this, but in actuality, they would look sort of akin to what's on the right here. So a slightly complex environment with two different phenotypes that are actually responding to each one of these environments. So on top of these two environments that are actually driving genotypic change, we also included a couple of sort of red herring environments, two environments that are correlated with, but not actually driving any type of evolution in these populations. And then we also included just 10 random environmental gradients to see if we could really break this thing. And then what we did was picking out an individual after we ran the simulation from one deme in the middle, say, and placing it somewhere else. Could we actually compare the GF offset to the calculated fitness offset of making that move? So we know, because it's a simulation, how fit every individual in every deme is. So if you move that individual out of that deme into another deme, we can just recalculate how fit they would be once they were there. So if we compare that actual fitness offset, in this case we're calling it a common garden fitness offset, with the gradient forest genetic offset, do we see a strong correlation to that? Do we see a strong correlation with that fitness offset to other measures, say environmental distance, either Euclidean or Mahalanobis distance, and to FST as well? And we also wanted to take a look at if we compare individuals within the core versus individuals on the edges, do we see a different type of effect based on more complex metapopulation setups? So we predicted that probably GF offset would maybe have some relationship to fitness, and the strength would vary based on location, and for how well it would perform compared to environmental distance and FST, we presumed that maybe it would do better. We were hoping it would do better. At least some of the authors were really hoping it would do better. Others were really hoping it wouldn't, because we wanted to actually challenge this. But what we ended up seeing, this is now a bar plot of Spearman's correlation coefficients between the common garden fitness and the GF offset. So if the GF offset was a really good predictor of how fit an individual that was locally adapted would do when moved to another environment, this value would be highly negative, because essentially the bigger the GF offset, the worse the fitness. And that's exactly what we saw. And whether we used all possible environments, including the red herring environments and the random environments, or filtered out all those environments to use just those that we knew were driving adaptation, we saw pretty good correlation between this measure of GF offset and fitness offset. We also compared this using environmental distance measures, and when we knew exactly what environments were driving adaptation, environmental distance performed just as well, if not better, than gradient forest did. And when we looked at FST, they were okay, but not as good. So we played around with this a little bit, and we attempted to constrain a couple of different environments to each other and see how well it would do. And overall, the GF offset was broadly very highly correlated with fitness offset. And interestingly enough, when we changed things around a little bit, because in all of the preceding examples I've been showing, we had two linear environments. And this is pretty much the baseline for how we can attempt to do this, is looking at linear environmental gradients and seeing how things change across them. But in this case, on the bottom here, the case 2 study, we actually employed non-monotonic environments, so essentially non-linear environments laid on top of each other, to see if when an environment is not perfectly gradient across our measured ecosystem, can we still hope to detect any level of environmental association here? And this signal definitely took a hit, especially when we were using all possible environments to inform the GF offset. But overall, we ended up seeing still a very highly correlated measure of GF offset to fitness. But again, if we know what environments are actually driving local adaptation, that performs way better in an environment where there is not just a linear gradient that explains it all. And as you can see, FST was not performing particularly well here. They performed about as well as if we were using a series of random environmental data. So the takeaway here is that gradient forest generally has a strong correlation to fitness offset, as we used in this very idealized model with very high local adaptation. And generally, it outperformed other what we called naive measures of offset, so FST or environmental distance, except when the environment is not perfectly linear. Then environmental distance, once you know which ones are actually driving adaptation, performed the best. So to wrap up, it's very likely that this will be a future we have to contend with. So knowing that 2.6 RCP has essentially been abandoned now as a potential goal, 4.5 is our current best case scenario, but I think it's very important for anybody working in marine environments to fully acknowledge and prepare for a potential future where we're contending with 95% environmental displacement. And in order to do that effectively, we need to have a very good baseline, not just of techniques and tools, but data. And the type of data that we need is going to be critical. One positive aspect here that I wanted to touch on very quickly is this idea of we understand quite a bit about adaptive potential based on effective population sizes and sort of how we can kind of measure how likely a population is to be able to respond to something as dramatic as this. And there's always more and more reasons to think that the individual genomes might be able to adapt to different scenarios in different ways than we've seen before. So I just wanted to highlight very quickly here at the end this concept of genotypic redundancy. So to explain this very quickly, just from looking at genotypes here, A, B, C, and D, these are different genotypes within individual animals that are all producing the same phenotype. So in the column phenotype, the little blue circle with the big black bar, those are certain phenotypes. And different genotypes within a population could all be segregating simultaneously, but perhaps with various fitness effects. So in this case, A and B both produce the same exact phenotype, and they have just about equal costs. So they have the same overall fitness, as opposed to genotype C here, which perhaps due to a plyotropic trade-off, produces the same phenotype, but results in much less fitness. So even though it exists in the population, we're unlikely to see it persist over a long time. And there is the theoretical possibility that there is a genotype D that, if it did exist, would produce the same phenotype at a much higher fitness, but just evolution hasn't discovered it yet. So this type of redundancy thinking and talking about the genetic architecture that underlies these adaptations can be massively complex, and we're seeing more and more evidence for that every day. And so just regardless of the mutation rate, if there is the potential for genotypic redundancy, we would expect that locally adapted populations, when they begin to respond to new environments, are probably gonna be exhibiting some level of transient genotypic architecture. And the more transient that your architecture is capable of being, that is to say, the more overall redundancy is possible to exist within that genome, the greater your chances are to actually be able to form novel approaches to new environmental challenges. And just to highlight here a little bit of work with the Atlantic silversides that I've been doing with Nina Terkelsen at Cornell. When we're looking at genetic architecture, I was comparing before different genotypes across the genome. But if we actually look at inversions, which are large areas of the genome that have essentially moved around a little bit, they've reoriented themselves, we can actually start to compare different parts of the genome's sequence divergence to closely related species. And in this case, if we look at non-inverted regions, we see that Minidia, the Atlantic silversides here in the middle, are compared to, two of the same species, pretty similar to each other and much more distinct from two other silverside species, as opposed to within an inversion where the sequence divergence within a population is actually much closer to interspecies divergence. So this type of genetic variability within populations can potentially be a hotbed of evolutionary potential, which isn't as easily measurable by our current approaches of effective population size or simple diversity. So just to end here on a short and simple to-do list, for myself at least, and that would be that the three things that are critically important moving forward to see the future of marine life would be large-scale environmental data. And it's my understanding that Hafró is actually actively and with great plans to contribute to investing in huge data sets that we can use, not just by nearshore environments, but larger out-to-sea environments, data that's publicly available, that's usable by researchers to actually understand how things are shifting, and can be used by different modeling groups to see how likely these things are to stay within our sphere of influence. And molecular references. So this has been touched on already by ERGA and the Ocean Genome Legacy Center, where essentially we need to know what diversity is out there if we're to understand what we're losing. So being able to actually sequence as many things as we can, even if it's a single token sample of some organism that exists currently in the environment that we're looking at, is going to be critical. And in terms of monitoring and sampling in the future, I feel like every single study that you're collaborating with or that you are participating in needs to submit for posterity a extractable and sequenceable data tissue specimen so that we know, even if we're not actively using it right now, we have a storage bank of tissues that we can sequence and will be able to compile projected genetic data sets into the future with. All right. And on that note, I'd like to acknowledge several collaborators from across, at this point, it's a little bit US biased, but there's at least one representative from Canada, Dr. Sam Yeaman from Calgary, and Dr. Snæbjørn Pálsson at the University of Iceland. But on that note, thank you for your patience and your attention, and I'd be happy to take any questions.
I think it's on, right? Yes. Thank you so much for this talk. It was super interesting, and it's a very novel kind of approach, at least for me. I've been working on climate change and ocean acidification, but to get this genetic component into that is quite important, I think. Are there any questions in the room? Yeah, I think
Very fair.
So I think a lot of what you presented is kind of new for, at least for me. So I think a lot of people here are processing this and trying to understand how we can kind of approach this holistically. At least I am trying to do that.
Right.
Can I maybe do you have a question?
I have many questions, but you can.
Hi, tack.
Hi.
I'm wondering about because there might be some parts of the population that get through it, some response that we haven't seen before, but this is definitely going to have a big bottleneck, right? And
So the idea is that we all know that not just worst case scenario, but just what is going to happen is that there is going to be a lot of change very quickly. And if there is not a lot of adaptability, a lot of things will go extinct. How much adaptability is actually present is really hard to properly gauge. But there is a possibility that there are novel ways of being adaptable that we don't fully see yet, or we don't have an easy way of measuring just yet. One of the things that Sam Yeaman has been working on especially is this idea of a C-score, which is a way of measuring redundancy, and it's very time-intensive and it's very expensive because you essentially just have to rear multiple populations in the same environment multiple times and sequence them all at different time points. Which is great, and you can actually start to get some quantitative measures at that point of how redundant is the genome. And it does seem to be pretty highly correlated, at least in simulation work, with redundancy and potential novel ways of approaching different environmental problems. However, we just don't have the empirical data yet that we need to catch up with these ideas and these theories. So once we have sort of more of that empirical data to start to compare, we can do a better job of assessing just how likely some populations are to adapt or not to adapt. Because so far, mostly what we've gone with is variation within a population. So like the coelacanth, it's an evolutionary dead end. It doesn't look like there's a lot of diversity. It's a very old lineage. There's probably no future for it. Well, who knows? It's made it this far. Perhaps there's something underlying in its genome that in a different environment would actually allow it to precipitate into a new setting. But yeah, so I'll say that right now, yes, it does look like it's just going to have to be a lot of bottleneck if we truly know everything there is to know about adaptability. But if marine life can still surprise us and there are different ways to adapt to it, then I think it's quite likely that we would see not quite the level of mass extinction that it currently looks like.
Yeah, one question. You mentioned earlier on that there have been within a fossil record, there have been a major climate events. You mentioned there's been, I can't remember when, but big dropping temperature. Mm-hmm. Can we use the fossil records to give a clues about potential, genetic adaptability in the future? I mean, is there something from there, say, these type of animals, they died and they didn't respond to this manner and say, okay, this could be, at that time you mentioned it's cooling, this is warming. So mm-hmm. Is there anything we use there?
So that's exactly what I was hoping to do, was to see, like, are there clear signatures that there was strong selective pressure? The problem was that the data set that I had was all mitochondrial data, which is maybe not necessarily the best type of data to be looking for responses to changes in environmental temperature. Mm-hmm. But I think if we can see back in time that this particular superfamily of sea urchins made it through a pretty tumultuous period of Earth's story without really strong signatures of selection. Okay. That's perhaps could be interpreted as like maybe a bit of good news. Mm-hmm. That maybe there was a possibility that, at least from a mitochondrial perspective, there was not a dire need to have a full sort of free-for-all, quick find a new niche and spread out in response to this changing environment, and that those few individuals that survived were able to radiate in the future. But rather that, even though the mitochondria has a very, to quote Mike Whitlock, an incomplete natural history, there is some information there that maybe things can survive these types of changes over long periods of time without essentially a full change in their genomic makeup. The problem, obviously, is that the Eocene and Oligocene cooling took 10 to 20 million years. Mm-hmm. So it's really an issue of scale. And while urchins are amazing at reproducing and really just fantastic model organism, they might have come out of that a little bit easier than maybe slowly reproducing larger, say, mammals at the time. So I think while there's always a reason to be optimistic and to look for the positive angles on things, it's not a very clear-cut story either that is definitely a positive signal. But I commend you for actually seeing it there. And yeah, especially because a lot of this, I went through a lot of material very quickly. So my contact information is up there, my email, and I am using the Twitter handle to actually communicate with people as well. So please do feel free to reach out and talk to me more if you have questions in the future or need clarification on anything I've said today.
Sounds good. I will definitely do that myself. Thank you so much, Áki, for this talk, and hope we can at least collaborate in the future, if not get you on board here.
Well,
Thank you.
Thank you very much.